Okay, so I hope everyone had a restful evening yesterday. Welcome to day 2 of the Introduction to Single-Cell RNA-Seq workshop. Today we'll be talking about the process that takes your count by cell data from the sequencing machine and tries to prepare the data for the cell clustering step. So this is a continuation of our discussion about how to deal with variation in single-cell RNA-seq data. Single-cell RNA-seq data has an extremely high dimensionality, meaning there are often tens of thousands of observations or cells that each have tens of thousands of variables or genes. And the high dimensionality and sparseness of the data can be problematic for clustering algorithms. So researchers are often motivated to reduce the dimensionality and remove axes of technical variation prior to cell type clustering. So after we obtain our cell by gene expression matrix from the sequencing machine, people often attempt to refine their data using many filtering and dimensionality reduction tools. And the goal of this procedure is to prepare the single-cell RNA-seq data for cell type clustering. So yesterday we talked about the pre-processing, quality control, and normalization steps. And today we're going to go over dimensionality reduction approaches, including feature selection and principal component analysis. And the idea here is to find the biologically varying genes and then the axes of maximal variation before performing clustering to find transcriptionally similar groups of cells. We'll also discuss two common approaches to visualizing heterogeneity, which are the t-SNE and UMAP algorithms. And finally, we'll do another demo in ScanPy where we will quickly recap the quality control steps and then apply our dimensionality reduction tools and then do some more open-ended exercises where you can try to implement some of ScanPy's functions on your own. Okay, so let's get into it. So a general challenge in studies with big data, including single-cell RNA-seq data, is how can we comprehend data that has tens of thousands of dimensions? If we just look at a heat map of single-cell RNA-seq data in which each row is a gene and each column is a cell, and the block is colored by the UMI count for each gene, it's difficult to derive any hypotheses about gene expression patterns in the cells we are studying. So often clustering algorithms are applied, and these algorithms partition cells with similar gene expression patterns into groups. And then the groups are compared against each other for patterns of change. However, clustering itself on extremely high dimensional data is problematic. And this is known as the curse of dimensionality. The curse of dimensionality means that when you have data that is dissimilar in many ways, distances between similar groups of cells become smaller, such that it is no longer obvious how to partition the data into different groups. So if you have 20,000 genes and each gene's value varies across each cell, is there a way to identify which subset of genes contribute the most to the distribution of cells on this manifold of gene expression? So how do we know what is the most informative subset of genes for cell clustering and downstream analysis? And is there a way to estimate what is the most appropriate representation of single cell data for these tasks? And to be frank, we still don't really know. The steps to prepare single cell RNA-seq data for cell clustering are a very active area of research. And today I'll be presenting the standard method in the field for preparing single cell RNA-seq data for cell type clustering. However, I think the field desperately needs to better validate these steps. And generally, it would be useful to generate data where each cell's physiology is somehow annotated prior to generating single cell RNA-seq data. But anyway, let's talk about how researchers typically go from collecting the raw data to clustering cell types and doing a quantitative analysis. So we start with our quality control filtered raw data, which can contain around 20,000 genes and tens and thousands of cells. And a common first step is to first try to distinguish technical noise from interesting biological variation and remove the technical noise. So because single-cell RNA-seq can be modeled as a Poisson process or process with counting error, we can model certain expectations for what the variation due to technical noise looks like. And researchers model variation on a per gene basis, measuring how the distribution of the gene count varies across the population of cells. So highly variable genes are those genes that are selected because they vary more than what would be expected based on a null model of technical noise. So after highly variable genes are identified, principal component analysis is often applied. And PCA is a dimensionality reduction technique that finds the directions of maximal variance in the data and linearly transforms the data such that the reduced dimensional space is a linear combination of the original space. So each component in PCA is determined by some combination of genes. And often cells are then clustered for transcriptional similarity using the PCA coordinates as the basis. And finally, UMAP and t-SNE projections can be applied to visualize differences in gene expression patterns in 2 dimensions across all the cells in the dataset. So again, the goal of dimensionality reduction in single-cell RNA-seq data is to prepare the data for cell type clustering. And cell type clustering is performed on a cell-by-cell distance matrix where the distances between cells represent the L2 or Euclidean distance between 2 points in an n-dimensional space. And here the dimensions would be the principal component vectors that each represent a linear combination of highly variable genes. So let's further unpack the concept of a distance matrix. If you have 2 cells and they express the exact same set of genes at the exact same levels, they would occupy the same position in gene expression space, such that the distance between these 2 cells is 0. Here, smaller values are colored in blue and represent groups of cells with similar expression patterns. However, if you have 2 cells that have very different set of genes expressed, they would occupy points in the gene expression space that are very far apart. So gene expression space is the n-dimensional space where each gene identified in your experiment defines one of the dimensions. So each cell is an n-dimensional vector where each gene's count defines the cell's coordinates. And the Euclidean distance between these 2 points gives you the distance between cells. So the distance is calculated between every pair of cells in your data. And this is the matrix we use to perform cell type clustering to identify groups of cells with similar gene expression patterns. So identifying which subset of genes and transformations to use that most appropriately represents cell biology is a very active area of research because the generation of this distance matrix is critical. It determines what kind of cell clusters you will see and what the differential expression between these groups of cells will be. So in other words, the clustering you achieve critically determines the results of your experiment. And the clustering you achieve is sensitive to all of the techniques that we apply. And as you can see, this whole process involves a lot of compressing and twisting and turning the raw data. And how well this process allows us to separate cells based on physiological differences in biology is still not well understood. So at this point, I'd consider this standard process more of an art than a science. So when you apply this to real data, the accuracy of the process is not guaranteed, and we should be critical when we interpret the results. And I think it's really important and helpful to understand how each analysis steps can affect your experimental results. But nonetheless, science progress is incremental. We're always able to go back and reanalyze our single-cell RNA-seq data using better methods or more refined approaches as they're produced. So anyway, the goal of feature selection and dimensionality reduction is to generate this cell-by-cell distance matrix upon which cell type clustering can be performed. So now we'll talk about each step in detail, starting with feature selection. And the goal of feature selection is to select the subset of genes which are most informative of biological differences across cells. So in the feature selection step, researchers often reduce the number of genes from 20,000 all the way down to 2,000 or so. And then they only use the top 2,000 highly variable genes for the principal component analysis and cell clustering step. So ideally, feature selection identifies those genes which are biologically variable, which means that the gene is differentially distributed across subsets of cells with different physiologies. And the idea of feature selection is to model technical variation and exclude genes whose variation can be explained by a null model of technical noise. So for example, here gene A has little variation across cells in the tissue, in this tissue cross-section. And as a result, this gene's distribution across our cells is fairly narrow, and its variance is relatively low. Specifically, this means that most of the UMI counts for this gene fall close to the mean UMI counts across all of the cells for this particular gene. In contrast, we can see that gene B has tissue-specific expression, where it's more highly expressed in these duct cells compared to the surrounding tissue. And when we quantify this using single-cell RNA sequencing, we would hope to see this differential distribution reflected in the distribution of this gene's UMI counts. So when we plot the specific mRNA gene counts in a histogram over all the cells in our sample, this is reflected. So in the feature selection process, we would expect that variation observed in gene A can be explained by a model of measurement error, whereas gene B is biologically varying, and the variance can't be explained by a null model of technical noise. So how can we build a model of technical variation? The standard approach seeks to identify highly variable genes, and this approach calculates the coefficient of variation for each gene, which is a measure of how spread out the gene count data is over all of the cells, relative to the mean count for that particular genes over all of the cells. So in other words, it's a measure of the standard deviation of a gene relative to its mean expression. And the idea is that the genes with the highest dispersion, or those with the most spread out values, are likely to be so dispersed because of real underlying biological variation. So in the highly variable gene selection procedure, we first calculate the coefficient of variation for each gene across a population of cells. And then we fit a generalized linear model to model the coefficient of variation relative to the mean. And this function attempts to model the variation that would be expected to arise from the capture process alone as a function of the starting concentration of the mRNA. So this model of technical noise can be parameterized using either the distribution of all of the genes or of synthetic ERCC spike-ins, which are colored in blue in this scatter plot. And because the ERCC spike-ins have a fixed starting concentration and are uniformly distributed throughout all of the droplets, the variation in the ERCC count should only arise from technical noise in the capture process and not due to any biological variation. So the current standard approaches advise selecting 2,000 to 5,000 highly variable genes for downstream analysis. And the Theus paper also recommends modifying the number of genes you choose based on what kind of downstream clustering results you see. And I hope that you can see how modifying the number of genes you choose can be problematic. And this arbitrary and potentially biased selection process can drastically change the results of your analysis. This is called data peaking, because you're peeking ahead at what the results should be and influencing what they are. Okay. So now we've talked about the first 5 standard steps in dealing with technical variation in preparation for cell type clustering. And so to briefly recap, these steps are using UMI counts to avoid amplification bias, using quality control metrics to filter out low quality or dead cells, normalizing the data to account for cell size differences, which may or may not, probably not affect the capture process. Then we do a log transformation to stabilize variance due to differences across in the mean. And then feature selection, which involves identifying the set of the most highly variable genes as a function of the mean. And then the final step in the standard pipeline is reducing the dimensionality of the data further using principal component analysis. So I will briefly explain principal component analysis, or PCA for short. And the goal of PCA is to find which linear combination of genes accounts for the most variance in the distribution of the cell gene data. And why do we need to do principal component analysis, you might ask? The idea is that by selecting the directions of maximal variation, the clustering algorithms will work better. This is thought to be because there are still so many zeros in the HVG space, which can potentially cause issues in finding groups of similar cells. So PCA is used to reduce the dimensionality of the data so that each cell is plotted in axes that represent directions of maximal variance. And these directions of variance are determined by principal components, which represent a kind of metagene, which is a linear combination of information across correlated sets of genes. So let's briefly go through PCA step by step. So first, imagine a scenario where we have 6 cells and 2 genes, and we plot the cells based on the UMI counts for gene 1 and gene 2. The cells appear to naturally fall into 2 groups, one cluster here and another here. And the first step in PCA is to mean center the data. This just shifts the points so that the center is now the origin. This does not change how the data are positioned relative to each other. And then the next thing that we do is a regression to find the line that minimizes the distance from all the points. So this line is called principal component 1. And to make the first principal component, we linearly combined the contributions of gene 1 and gene 2. That is, we took 0.97 parts of gene 1 and 0.24 parts of gene 2. And these proportions are the loading scores. And this line is also called the eigenvector. So to find the second principal component in a 2D graph, we find the line through the origin that is perpendicular or orthogonal to PC1. And then to calculate how much variance is explained by our principal components, we calculate the eigenvalues for each principal component. And the eigenvalue is the sum of the squared distances between the projected points and the origin. So we then take the squared sum of the distances for each PC and sum the total variation together to measure how much variation each PC accounts for. So in this example, if PC1 has a sum of squared differences of 15 and PC2 has a sum of squared differences of 3, it's either closer to PC1 than, or sorry, closer, they vary more along the axis of PC1 than PC2. We add them together and we can calculate the proportions to say that PC1 accounts for 83% of total variation around the PCs, and PC2 accounts for less. So we can then graph the proportion of variance explained for each PC to make a scree or an elbow plot. And this graph is useful for determining how many principal components we want to use in our distance matrix. So you can imagine if we extend this into 2,000 more dimensions, we just continue the process of fitting linear lines through the origin that are orthogonal to each other and calculating variance explained. And then after a certain number of dimensions, we often find a plateau in the variance explained, where adding additional PCs only contributes a minimal amount of variance explained. So this here is considered the elbow dimension, and it determines the number of PCs that we want to use in the distance matrix. And we can see that the first PCs tend to capture a large amount of the variance in our data. Okay. So after we perform quality control, normalization, feature selection for highly variable genes, and dimensionality reduction using PCA, we find the elbow dimension of our principal components and project our cells into a reduced coordinate space where each axis is a principal component that explains a high fraction of variance in the data. So the next step is calculating the cell by cell distances using this dimensionality reduced basis. Okay. But before we go on into our next segment, let's look at what PCA does to log transformed counts per million normalized data. I've showed you how the zeros in our gene by cell matrix can generate a lot of artificial generation or variation when CPM normalized. And this is a concern because PCA's job is to identify directions of variation. So this motivates a hypothesis that the fraction of zeros in UMI counts might influence the selection of PCs for our downstream analysis. Okay, so we can look at the data. Because Townsend and all have done this experiment for us. So again, this is a histogram of the UMI counts for a single gene across a set of monocytes. So there's only one cell type. And you can see that there is a high fraction of zeros in this particular gene counts, likely due to the low probability of capturing a not super abundant mRNA. But otherwise, this gamma distribution has relatively low coefficient of variation spread across its mean. So in contrast, when we do the CPM normalization, the zeros remain zero, and now many of these cells fall further away from the gene's mean. And if we take the log of the CPM plus 1, this moves the fraction of cells with zero counts even further away from the mean. So in the Townsend et al. Paper I shared with you, they sought to evaluate whether these transformations introduce bias into the standard single-cell RNA-Seq analysis pipeline. So they first performed the standard dimensionality reduction pipeline using a synthetic data set where single-cell RNA-Seq was performed on droplets containing only the synthesized ERCC spike-ins. The y-axis in the other data is the UMI count per gene. In this case, the transformed count value. So we start off just with the raw UMI count data, which is the sum of each unique molecular identified molecule for that particular gene species. And it's a histogram of what this gene's distribution looks like over a set of cells. So we're looking at one gene's counts over all the cells that we sequenced. And this is the raw data after quality control steps. This is the counts per million transform data that does nothing to zero values because a zero proportion is still a zero. And this is the log transformation of CPM counts plus 1 because you can't take the log of 0. And you can see now that the variance will be a lot larger because a larger proportion of cells falls further away from the mean of this distribution for this particular gene. So the Towns group wanted to evaluate how this transformation affects the standard analysis pipeline. So they first performed an experiment on data where there is no biological variation. They did the standard dimensionality reduction pipeline using the synthetic data set where single-cell RNA-Seq was performed on droplets containing only synthesized ERCC spike-in cDNAs, so there's no actual cells in the data. And after performing quality control filtering, CPM normalization, log plus 1 transformation, and PCA, they project the ERCC count per cell data onto the first principal component and plot it against the fraction of 0 counts for each droplet or simulated cell. So here the y-axis is the first principal component or the direction of maximal variance in the data. And the x-axis is the 0 fraction or the number of 0 entries for each particular cell. And each dot represents one droplet or fake cell represented in PCA space versus just the proportion of 0 counts per UMI. And what you find is this trend where as the droplets have a greater fraction of zero counts, they fall on a higher position on the first PC. So what this means is that principal component 1 is essentially capturing the zero fraction of the data and separating the droplets based on how many zeros they have. And this is concerning because it raises the possibility that the standard dimensionality reduction pipeline is separating cells based on technical variation rather than biological variation. So to test this, they did the same analysis, but on real data. But here the data is comprised of just one cell type. So there shouldn't be huge variation in smaller versus bigger cells that might have more or less counts of genes. The distribution in the fraction of zeros should be fairly uniform depending on the heterogeneity of this cell type. So here you can see how the first principal component captures the zero fraction again, or which cells have the highest proportion of zeros in their UMI count across all genes. So the higher fraction of zeros is indicated by blue. It can go in either direction. It's just distance. Okay. So this observation again raises the concern that the standard pipeline, which uses the CPM and log+1 transformation of highly variable genes, might be conflating technical variation with biological variation. However, how the zero fraction maps to biological function in real data with more diversity in cell types remains to be determined. So in other words, whether this normalized, transformed, and subsetted selection of genes is the most appropriate representation of differences in cell physiology is an open area of research. And again, to test this idea, we can try to generate more orthogonally annotated data sets where the physiological state or a phenotype of interest is known before sequencing. And then you can try to map which set of genes best represents differences in the physiology of your cells. And there are a few studies which try to consider this. The Towns paper is one of them, but it's a tricky problem and there are not very many annotated data sets available. And this is a major hurdle for progress in the field. Okay. So I'll take a pause here for questions if you have any.
I had a question a few slides back. You mentioned general linearized model using the gamma distribution for feature selection. Is that correct?
That's correct, yes.
And so in that, can you just explain to me one time what the dependent variable is and what the independent variables are in that equation?
So it's I don't know if I could almost give up. We're modeling what the how the coefficient of variation depends on the mean. So there's some expectations that the mean variance relationship is constant, which might not be right, but it's mostly looking for which genes have the highest variance depending on their mean.
So the dependent variable is sort of is it the coefficient of variation, and then the gene itself is the independent variable in that equation? Is there a site or something you could send me to look at that gamma generalized linear model? I just wanted to look at it a little bit closer.
Oh, sure. Sorry, I can't, my Zoom window is missing. Can you maybe send me an email or message?
Of course.
Thank you. Yeah, I can do that. And yeah, I think your intuition is correct. Okay. Okay, so for the elbow plot, is it best practice to take your number of PCs right at the elbow of the graph? So at that slide, I think it was about 5 or 6. And yes, this is recommended to take the number of PCs right at the elbow. Though in a recent work from the lab that I work in, we showed that this practice of reducing the dimensions of your data at the elbow might not be the best approach to preserving the topology of variation in gene expression space. But in general, people just visually inspect and look at the elbow and decide that is where we will cut off the number of dimensions that we use to project our data.
Sorry, could you repeat that last sentence you said, that it's, what is it due to the data?
Oh, so we did an experiment where we measured how much distortion was introduced into the neighborhood structure, which I will talk about a lot more right now. So the idea is comparing the cell neighbors in high dimensional space to the cell neighbors in the low dimensional PCA space to see whether or not the neighborhood structure is preserved after you reduce the dimensions using PCA. And what we found was that the elbow direction, it was kind of more of a continuous function of distortion. So the additional dimensions after the elbow dimension do tend to have important information about the high dimensional topology of your data. So cutting it off using only the largest PCs can potentially introduce distortion into your analysis. There's a loss of information about the topology.
Got it, thank you.
Okay. So today's lecture will be much shorter, and we'll end today with a discussion of ways to visualize variation in high dimensional data. And then tomorrow we'll go on to discuss cell type clustering. So UMAP and t-SNE projections are visualization tools that are typically applied to PCA transformed raw data or highly variable gene data. And the goal is to visualize whether cells with known biological markers, gene markers, cluster together in expected ways. So the purpose of these two-dimensionality reduction projections of the data is data exploration. They are absolutely not designed to create a basis for distance measures in the same way that PCA can be applied to count by cell data. So t-SNE and UMAP are nonlinear neighborhood projection algorithms that try to preserve pairwise distances between cells, but they are not designed to preserve relative distances in the neighborhood structure of the cell gene data. So essentially both t-SNE and UMAP use loss functions that make similar points in the cell by gene distance matrix attract each other, and it pushes away dissimilar cells from each other. So here's an example where we visualize two different neuronal markers, and each point is a cell, and the color red is used to indicate whether the gene is expressed within each particular cell. And as you can see, this t-SNE projection can be useful for visualizing how broadly marker genes are distributed across a population of cells, where this one's much more broadly expressed and absent in the cells that have this more locally or focally expressed EOMs gene. And then later on, we can then check to see whether these subsets of cells with marker gene expression map to the clusters we generate using the unsupervised clustering approaches. Oops, sorry. So t-SNE and UMAP are both dimensionality reduction algorithms that attempt to preserve pairwise distances between neighbors. And both algorithms rely on a k-nearest neighbor graph. And to construct a k-nearest neighbor graph, we first calculate the Euclidean distance between cells, which represents the cell-by-cell differences in gene expression space. And then we choose a k value. And K is the number of neighbors to identify. So in this case, let's say 20. So to construct the K nearest neighbor graph, we simply find the 20 closest neighbors of each cell based on our distance matrix in the high dimensional space, or whatever reduced dimensional space we're using. And the graph generates a neighborhood topology where we have structural information about which cells are neighbors with each other in gene expression space. So t-SNE stands for T-distributed stochastic neighbor embedding. And as the name implies, it is a dimensionality reduction algorithm that uses a random process to project the high dimensional data into 2 dimensions. So to construct a t-SNE plot, the first step in the algorithm is to initialize the data using PCA and then compute the neighborhood graph. And then local neighbors have attracting forces while dissimilar neighbors have repelling forces. And while t-SNE tends to do an okay job at describing the structure of very local pairwise similarities between cells, the global structure of the original data is not preserved, which is to say you cannot compare the distances of 2 points on this graph, which represent cells, and make inferences about how similar their gene expression profiles are, even though they look to be close, and it's kind of natural to think sometimes that maybe these cells differentiate into these cells and so on. t-SNE and UMAP really don't preserve anything about global neighborhood structures. They make a lot of unexpected slices through the data to project it down to 2D. What makes the images shaped the way they are? There's a lot of hyperparameters. Let me show you the next slide. Okay, so UMAP, it's similar to t-SNE in that it's another visualization technique that performs dimensionality reduction to achieve a uniform manifold approximation and projection. So UMAP aims to optimize the layout of the data representation in the 2-dimensional space by minimizing the cross entropy between the high dimensional and the low dimensional topological representations. So these representations are fuzzy topological sets of the data that UMAP uses to approximate a manifold on which the data lies, and then construct a fuzzy set representation of the approximated manifold in 2 dimensions. So reconstructing a high dimensional manifold in 2 dimensions always induces distortion into the neighborhood structure of the data. That is to say, again, UMAP is a visualization tool, and it's not appropriate to use UMAP projections to infer how similar different cells are in relation to each other, because a 2D projection is an approximation that induces an extremely high level of neighborhood distortion. What we think is happening is that in the dimensionality reduction process, we have to slice regions that are connected in high dimensional space to project them onto this graph in 2D space. So while the clusters may look far apart here, they might actually be really close in the high dimensional space. So again, it's not appropriate to use UMAP coordinates for downstream analysis, even though it is unfortunately kind of a common trend and very common to talk about the data like it is preserving some sort of structure in presentations. And research in the Deeds lab has found that if you compare cell neighbors in high dimensional space on average, compared to this low dimensional projection, 98% of the neighborhood structure is totally different when you compare high dimensional neighborhoods to the neighborhoods found in t-SNE or UMAP spaces. So that is to say, t-SNE and UMAP almost completely distort the topological cell neighborhood structure. Neighbors meaning close in gene expression space, yes. Meaning they have similar sets of genes expressed in similar quantities. Okay, so both of these tools have hyperparameters that are illustrated here. And these control kind of what the two-dimensional projection looks like. So across the columns of the graph, we are titrating the number of neighbors used in the UMAP projection. The number of neighbors informs the algorithm of how many neighbors you want to consider when attempting to approximate local neighborhoods. So in other words, it's a parameter that modulates the granularity of the projection. Do you want to look at the whole world of neighbors, then you would use a larger K value. But if you only want to look at like your local neighborhoods on a street in gene expression space, you would want to use a smaller number of neighbors. And across the questions, each t-SNE UMAP represents a single sample. Yeah, so in the t-SNE and UMAP projections, each dot is one cell. And instead of plotting each cell in a 20-dimensional, well, you can't plot things in a computer in 20-dimensional space and look at it. So instead, what people do is reduce the 20-dimensional coordinate points down into two, an X and Y coordinate system determined by UMAP or t-SNE. And so each dot here represents a cell and all of its genes collapsed into 2D space. Okay. So they don't represent treatment or not treatment. You can do this when you have treated on treatment groups, if you pull all the data together, or you can do it separately. In general, they're used for plotting like known marker genes and then seeing how that corresponds to your cell type clusters. So you can label the cells according to the expression of a gene of interest or what cell type cluster they belong to, and then kind of compare the overlay of that visually to have a sense of what your clustering algorithm is doing. Gene expression space is the n-dimensional vector where each cell is a vector space and each dimension is defined by one gene. So you imagine you just have a string of numbers that provide the coordinates of each cell according to the values of each gene you detect. So one cell, and then you have 20,000 columns where each column represents the UMI count for a particular gene. So gene expression space is the space or region defined by these 20,000 vectors. And then you can plot cells as coordinates within the space and compare how close they are to each other. So some issues with doing this in 20,000 spaces is because there's so many variables, it's such high dimension. Distances between similar cells and different cells become smaller. So people often reduce the dimensionality of the data to only include the most highly directions of variability so that the distances between different cells become bigger. So clustering is essentially arbitrary. Yes, you will always find clusters no matter what. The job of a clustering algorithm is to find clusters. Whether or not they're appropriate is hard to say. I read most of single-cell RNA-seq analysis papers with a very high level of skepticism in what they say. There's useful ways to kind of evaluate clusters, but the best approach is always to have some orthogonally labeled data so you can at least compare your clusters to some historical notion of what cell identity is and maybe find some unexpected differences in what the field says about cells expressing X gene versus the observed heterogeneity within them. Okay, lots of questions. Okay, PCA, yes. Typically, you can apply t-SNE or UMAP to whatever data you'd like. So you can apply the t-SNE and UMAP to look at your raw data and compare it to the PCA reduced data and see if they give you similar representations of your data. Not sure I really understand the question. So if you want to clarify, you can send another message. I'm going to move on to the next one. So Harry asks, is it actually possible to perform nonlinear dimensionality reductions that preserve neighborhoods where neighbors are defined by linear Euclidean distance metrics in gene expression space? And absolutely, yes, that's one of the major projects that Seamus is working on in the DEEDS lab. So they're working on constructing a manifold learning technique where the objective function preserves the average Jacquard distance, which is a metric of the difference between the overlap and the union of the high dimensional and low dimensional neighborhoods. You might recall seeing Seamus give talks at QC bio seminars. But also I can send a link later on to the preprint that describes this approach. Actually, there's not a preprint, but we've used this metric in other ways to describe how much distortion is induced by different techniques. Okay, so the next question Ozzy asks is, hopefully we'll have workout soon, but what if treatment triplicates versus untreatment triplicates, each single sample have their own t-SNE UMAP, then how to compare DEG? So we'll talk about that. So the point I'm trying to convey here is that t-SNE and UMAP are only used for visualization, just to give some intuition about what the distribution of genes looks like across your samples. It's really not appropriate to use them as a basis for any quantitative analysis. So to compare differential gene expression across groups, that's often done in the raw gene expression space. The purpose of everything I've talked about today is to prepare the data for cell type clustering. But once we've found the cell type clusters, we annotate the cells. So we index them according to which cluster they belong to. And then people would typically, if you're interested in, say, like the stem cells across different treatment groups, you would compare the differential gene expression of the two stem cell identified clusters in the raw dimension space. So you would use the raw count data to do the differential gene expression analysis. But everything we're talking about today is just to prepare the data for clustering. We don't actually use transformed counts when we make quantitative measurements of gene expression. Cool. So do we apply t-SNE and UMAP to the output of PCA, or is it better to apply directly to the raw data? So this might be a little bit tricky to explain because t-SNE and UMAP both use PCA to initialize the data, which means they just project the data into its axes of variation to start with. So you can input the raw data, or you can input the feature selection data, or you can also input the PCA reduced projection where you only feed it the first 20 principal components. But it uses principal component analysis to kind of give an initial embedding of where the points should fall before reducing it further to 2 dimensions. Okay, cool. Thank you for all the excellent questions. Okay. So another way you can change what the UMAP looks like is by titrating the minimum distance that's used in the UMAP algorithm. So across the rows in this graph here, we're titrating the minimum distance, which is a parameter used in manifold approximation. It's used to decide whether or not cells are close enough in gene expression space to be neighbors. And you can see how this gives slightly different visualizations, but overall the pattern's kind of similar. Okay. So I have a few more slides that goes a bit further and illustrates the idea of distortion in dimensionality reduction. So here I will plot the map of the Earth using 3-dimensionality approaches that we have discussed so far. So the first panel here illustrates a synthesized set of coordinates for continents on this planet Earth, laid out in 2 dimensions. The second panel is a 2D projection of the data after applying PCA. You can see that though the points are rotated, the neighborhood structure is retained because we are embedding a 2-dimensional manifold into a 2-dimensional space. And principal component analysis preserves all of the points' relative location to each other. However, when we apply t-SNE or UMAP to the same data, we do not achieve the same global distance preserving effect. t-SNE is designed to retain pairwise distance relationships, while UMAP projects fuzzy topological neighborhood representations. And both use nonlinear approaches and are not designed to prioritize the preservation of neighborhood structure. So next, if we instead try the more difficult problem of projecting a 3-dimensional manifold into a 2-dimensional space, something very different happens. This is because PCA, as a linear algorithm that assigns equal weights to all pairwise distances, cannot preserve the structure. While in contrast, non-linear manifold learners such as t-SNE and UMAP prioritize distances between neighbors. So in other words, t-SNE and UMAP can compute something about neighborhood structure of all of the data, while PCA cannot. And by prioritizing certain neighborhood attributes instead of treating all relations as equal, t-SNE and UMAP are able to better represent 3-dimensional data in 2-dimensional space. So as you can see, a fair amount of distortion is introduced. So this is why it's important to use an appropriate number of principal components when embedding your data using PCA. You want it to be at least the latent dimensionality of your data, so ideally 3, not 2. Okay. So here's the most recent publication on these visualization algorithms, and it shows what happens to a toy data set of a circle when different initializations are applied to the embedding algorithms. And here the points were sampled from a circle with a small amount of Gaussian noise. For t-SNE, either random or PCA initialization was applied to the data before embedding, and UMAP used either random or a technique called Laplacian eigenmaps to initialize the embedding. And again, both tools work by using a loss function that makes similar points attract each other and pushes away dissimilar points. So the loss function is minimized using a gradient descent algorithm, which begins with some kind of initial configuration of points in space. And through iterations, points that are moved around in a way that decreases the loss function. So it tries to preserve similar neighbor cells, or in this case, circle data. And the initial configuration is decided by this initialization function. If you have a random function, t-SNE does not do so great. Points are projected randomly onto the space, and neither t-SNE or UMAP can recover the manifold of the circle. t-SNE produces a knot, while UMAP produces this tangled web with many, many tears. So in contrast with informed initialization, both t-SNE and UMAP can recover this manifold that resembles a circle. Though as you can see in this simple case, neither very accurately preserve the topology of the circle in the embedding. Okay. All of this is to reinforce the point that visualization is an essential and useful tool for data exploration, but the coordinates of the data in 2D space cannot be used to infer how similar distance cells are to each other in the gene expression space. So if anyone has any more questions, I can take them now. I also found recently an interesting video on StatQuest about how t-SNE works. And if people want to watch it, I can play it now. It's about 10 minutes long. And if you don't want to watch it, maybe you can take a break. Maybe vote in the chat, please. This is all the slides I have prepared for today. Diffusion maps are interesting idea to practice, but I would need to refresh my memory on specifically what they do. I think they have more useful uses in particular contexts, but I'm blanking right now on how it specifically works. But good question.
Brianne, I have a question about the principal component analysis again. So basically, if you have like 20,000 genes, you are collapsing the sort of distance between pairwise every two genes down into one principal component, right?
In one principal component, it is a linear transformation of all the genes interactions, yes, but typically people use more than one principal component. Like typically it's around 20 or so, which are kind of all meta-genes.
So, in the example you gave previously, the principal component was only like PCA1 that takes into consideration all of those gene-gene interactions.
Yes.
I was wondering in that example, what was principal component 2?
That is a great question, and we could potentially work that into our exercise today. I don't know off of the top of my head what it was, but I'll show you later on how we can look at what the loading components are of different principal components, so you can see how much each gene contributes to them. But in general, it's a good practice to kind of break down the components if you'd like to look at whether or not other like quality control metrics correlate to the loading scores, like whether like you have total number of UMAP counts or fraction of zeros. And stuff
What
like
do I see? What do people normally use as the principal component 1 and principal component 2 when doing these analysis?
So it's an unsupervised manifold learning technique, principal component analysis. So you don't tell the algorithm which combinations of cells to use, or sorry, genes to use, but it projects the data and finds the covariates that show you which directions or which combinations of genes produces the greatest directions of variation. So I've showed you what people normally do, which includes the whole normalization, transformation, feature selection, dimensionality reduction approach, and then they apply principal component analysis to the normalized transformed HVG space. And then to do the clustering analysis, they typically take all of the principal components up to the elbow dimension, and then separate cells based on their coordinates in the projected PCA space. And often people don't really look at what the loadings of the PCs are, but it can be informative to look at and see if whether there's a group of genes contributing to the loadings or whether it's just dominated by like one gene that might be an outlier for some reason, just to give you an idea of how you're biasing your cell type clusters.
Okay, thanks.
Yeah. People have explored other distance measures, and it's a good question. There's no biological reason why we use the L2 space, although it is like a nice mathematical number where it gives us like stable behavior in a lot of different variance estimates and stuff. So mathematically, it's a nice number. Whether it's like the most appropriate representation of biology, who knows? No one knows. But generally, the L2 distance is well behaved, so people tend to use that. We have tried using other distance matrices and kind of mapping it to annotated data, and we find kind of unclear results that it doesn't seem, we don't have any reasons to use other ones, I'll say. They weren't very compelling. Okay, does anyone want to watch this video about t-SNE, or is everyone gone? Okay, I'll put it on. I'm not sure necessarily if I can figure out how to get the sound to play. Maybe if I unplug my headphones. Does anyone know about that?
I'm drawing a graph. Doesn't it look cool? But I didn't know how it worked until I watched StatQuest. Hello, and welcome to StatQuest. StatQuest is brought to you by the friendly folks in the genetics department at the University of North Carolina at Chapel Hill. Today we're going to be talking about t-SNE, or t-SNE. To be honest, I don't actually know how it's pronounced, but it's going to be clearly explained, I know that bit. Also, this StatQuest is by request. A couple of people put it in the comments below, and I got a couple of emails from other people, so I'm doing it because you guys want it. Here it goes. If you're watching this StatQuest, chances are you've seen an example of a t-SNE graph before. What t-SNE does is it takes a high-dimensional data set and reduces it to a low-dimensional graph that retains a lot of the original information. If you're not familiar with those terms of taking a high-dimensional data set and reducing it to a low-dimensional graph, you might want to watch the StatQuest for PCA because I explain what that means in that video. Here's a basic 2D scatter plot. Let's do a walkthrough of how t-SNE would transform this graph into a flat, one-dimensional plot on a number line. I'm going to use this super simple example to explain the concepts behind t-SNE so that when you see it applied to a much larger data set, a much more complex data set, you'll still know how that graph was drawn. Note, if we just projected the data onto one of the axes, we just get a big mess that doesn't preserve the original clustering. If we project it onto the y-axis, instead of two distinct clusters, we just see a mishmash. And the same thing happens if we just project the data onto the x-axis. What t-SNE does is find a way to project data into a low-dimensional space, in this case, the one-dimensional number line, so that the clustering in the high-dimensional space, in this case, the two-dimensional scatter plot, is preserved. So let's step through the basic ideas of how t-SNE does this. We'll start with the original scatter plot. Then we'll put the points on the number line in a random order. From here on out, t-SNE moves these points a little bit at a time until it has clustered them. Let's figure out where to move this first point. Should it move a little to the left or a little to the right? Because it is part of this cluster in the two-dimensional scatter plot, it wants to move closer to these points. But at the same time, these points are far away in the scatter plot, so they push back. These points attract, while these points repel. In this case, the attraction is strongest, so the point moves a little to the right. Bam! Now let's move this point a little bit. These points attract because they are close to each other in the two-dimensional scatter plot. And this point repels a little bit because it is far from the point in the two-dimensional scatter plot. So it moves a little closer to the other orange points. Double bam! At each step, a point on the line is attracted to points it is near in the scatter plot and repelled by points it is far from. Triple bam! Now that we've seen what t-SNE tries to do, let's dive into the nitty-gritty details of how it does what it does. Step 1. Determine the similarity of all the points in the scatter plot. For this example, let's focus on determining the similarities between this point and all of the other points. First, measure the distance between 2 points. Then plot that distance on a normal curve that is centered on the point of interest. Lastly, draw a line from the point to the curve. The length of that line is the unscaled similarity. I made that terminology up, but it'll make sense in just a bit, so hold on. Now we calculate the unscaled similarity for this pair of points. Now we calculate the unscaled similarity for this pair of points. And now we calculate the unscaled similarity for this pair of points, etc., etc., etc. Using a normal distribution means that distant points have very low similarity values, and close points have high similarity values. Ultimately, we measure the distances between all of the points and the point of interest. Then plot them on a normal curve, and then measure the distances from the points to the curve to get the unscaled similarity scores with respect to the point of interest. The next step is to scale the unscaled similarities so that they add up to 1. Why do the similarity scores need to add up to 1? It has to do with something I didn't tell you earlier. And to illustrate the concept, I need to add a cluster that is half as dense as the others. The width of the normal curve depends on the density of data near the point of interest. Less dense regions have wider curves. So if these points have half the density as these points, and this curve is half as wide as this curve, then scaling the similarity scores will make them the same for both clusters. Here's an example where I've worked out the math. This curve has a standard deviation equal to 1. These are the unscaled similarity values. This curve has a standard deviation equal to 2. These points are twice as far from the middle. The unscaled similarity values are half of the other ones. To scale the similarity scores so that they sum to 1,
You take a score and you divide it by the sum of all the scores. That equals the scaled score. Here's how the math works out when the distribution has a standard deviation equals to 1. We get 0.82 and 0.18 as the scaled similarity scores. And here's the math for when everything is spread out twice as much. We get 0.82 and 0.18. The similarity scores on top are equal to the similarity scores below. They're the same. That implies that the scaled similarity scores for this relatively tight cluster are the same for this relatively loose cluster. The reality is a little more complicated, but only slightly. t-SNE has a perplexity parameter equal to the expected density around each point, and that comes into play, but these clusters are still more similar than you might expect. Now back to the original scatter plot. We've calculated similarity scores for this point. Now we do it for this point. And we do it for all the points. One last thing, and the scatter plot will be all set with similarity scores. Because the width of the distribution is based on the density of the surrounding data points, the similarity score for this node might not be the same as the similarity to this node. So t-SNE just averages the two similarity scores from the two directions. No big deal. Ultimately, you end up with a matrix of similarity scores. Each row and column represents the similarity scores calculated from that point of interest. Red equals high similarity and white equals low similarity. I've drawn the similarity from a point of interest to itself as dark red. However, it doesn't really make sense to say that a point is similar to itself because that doesn't help the clustering. So t-SNE actually defines that similarity as 0. Hooray! We're done calculating similarity scores for the scatter plot. Now we randomly project the data onto the number line and calculate similarity scores for the points on the number line. Just like before, that means picking a point, measuring a distance, and lastly, drawing a line from the point to a curve. However, this time we're using a t-distribution. A t-distribution is a lot like a normal distribution, except the t isn't as tall in the middle, and the tails are taller on the ends. The t-distribution is the t in t-SNE. We'll talk about why the t-distribution is used in a little bit. So, using a t-distribution, we calculate unscaled similarity scores for all the points and then scale them like before. Like before, we end up with a matrix of similarity scores, but this matrix is a mess compared to the original matrix. The goal of moving this point is we want to make this row look like this row. t-SNE moves the points a little bit at a time, and at each step, it chooses a direction that makes the matrix on the left more like the matrix on the right. It uses small steps because it's a little bit like a chess game and can't be solved all at once. Instead, it goes one move at a time. Bam! Now to finally tell you why the t-distribution is used. Without it, the clusters would all clump up in the middle and be harder to see. Triple bam! And now we know how t-SNE works. I've used a really simple example here, but the concepts are the exact same for more complicated data sets. Hooray! We've made it to the end of another exciting stat quest. If you like this stat quest and want to see more like it, please subscribe. And if you have any ideas for future stat quests, just put them in the comments below. Until next time, quest on.
Oh, that guy talks a lot slower than me. Oops. Okay, I hope that was useful. Why don't we take a 6-minute break and then start again at 2:45. Sorry, 2:55.
Installed again,
So
besides the one.
these two, we need to install scikit-learn, which you can do by typing this in the terminal. And we'll also need to install need. And hopefully everyone was able to get the material from day 1 to run without errors. Specifically, you would have to had changed the script I provided so that your directory matches the folder where you put the MTX and barcode files. So this path points to my computer's location, but you need to change it. Okay. So yes. Okay, while people are doing that, anyway, when it's done, you can load the libraries and hopefully see that they worked. And in the meantime, I wanted to show you just a visual of what the ScanPy annotated data object kind of looks like and how it stores the data in a format. Oops. So I'm sorry, but sometimes my Zoom while I'm screen sharing doesn't let me see the chat. And I saw there was a chat, but I can't see it. Okay, so anyway, the annotated data object is the format developed by ScanPy to store the UMI count by cell matrix, and also with alongside other annotations that we might add to the object while we're doing the analysis. So the main data is stored in something that you can access by typing anddata.x, and this is the data matrix that has our observation times variable values. So here, the observations are cells and the variables are genes. And if we wanted to say sum how many genes were identified within each cell, and we call that total number of genes, that annotation of variable would be accessible if we typed anddata.var. And that is where we can name things that are annotations of our variables. And in the other dimension, annotations, oh, sorry, I got that backwards. Annotations across cells are in .obs, and you can call whatever you named it as annotated and data.obs and then call the variable that we name. And then there's also some unstructured data that exists in a Python data structure called a dictionary. All the basis of these are pandas data frames, and the larger data file uses a SciPy sparse matrix to make the performance go a little bit faster. So today I chose to use a data set that only has 3,000 cells so that people could run it on their computers, but you'll often find with larger data sets, you can run out of memory pretty quickly and require this kind of sparse matrix format to do computations on your data. Okay, so hopefully everyone was able to get scikit-learn and need installed. Today we'll go through the quality control steps again briefly, and then we'll do all of the steps that are required in the standard pipeline prior to clustering or cell types. And then afterwards, I will split you into small breakup groups and challenge you to do some coding and analysis on your own. So please feel free to interrupt me as I go through this and follow along if you can. So I'm choosing to run this line by line. So I just highlighted the libraries and can import them. If you right click, you can see the command. For PyCharm, it's Control-Shift-Enter. So I'll go through this. Oh wait, Control-Shift-E. Alt-Shift-E. Here, we're going to load the data into our annotated data object again. We will annotate the mitochondrial genes to identify the fraction of mitochondrial genes, apply basic filtering to remove those sparsely expressed genes and low quality cells. And then as we go along this process, we're going to create copies of our data so that later on we can compare differences as we go through each step in some of the activities. So, so the first one you'll notice is QC data, which we can make a copy of the annotated object using this copy command. And we'll do the filtering on this QC data. So we will slice the annotated data objects to only include those observations where the number of genes are less than 22,000 in order to exclude potential doublets from our data. And we're only going to include those cell observations again, where the percent of mitochondrial counts are less than 5. And the idea there is to exclude those cells that are low quality or potentially dead fragments of themselves. Okay. So that should bring us up to speed for the new material, and we're going to create another different object called normalized data by creating a copy of the QC data. And here, again, we just use this Python annotation of function module or library module function to call the normalize command. And here we're going to, instead of using 1 million, just use 10,000. So we're normalizing to 10,000 reads per cell. And then we'll apply a log+1 transformation, which is again almost too easy to do using ScanPy just by calling all of these already existing functions. So when we want to identify the highly variable genes, there do exist some parameters that you can play around with. I think the idea to have a minimum mean here is to exclude those genes that are super variable, just by nature of having means that are very, very close to zero. And then we can plot using the plotting module what these highly variable genes look like relative to the rest. So here we can compare normalized versus not normalized, and we see that our normalization, which is the input for our model of highly variable gene, is kind of oversampling these genes down here that tend to have means closer to zero. So if you want, I suppose you could adjust the minimum of the mean in the threshold earlier, but depending on what assumptions you think are real and not, this is kind of the recommended way to go by these people. Okay, so now we're just going to freeze this data frame again by making an HVG data before we do the principal component analysis. So to do this, I'm creating a new observation of variables that are just the highly variable genes in our data object. And we'll apply PCA. So this creates a new matrix for which we can apply PCA to. But before we do that, we will scale our data so that each gene has the same variance across as its mean. Just kind of a standard step in PCA, which if you don't know that much about, I recommend reading. All right, so now we can reduce the dimensionality of our data using principal component analysis and visualize what our highly variable genes look like in the principal component space. So since this is a 2D screen, we're just going to use the first two principal components to visualize the data. And we're going to color it by this gene CST3, which has some significance in immunology, which I forget. And you can see when we scatter our data by the first two principal components, we have three kind of distinct populations where one of them is more enriched for this gene CST3, which is probably encouraging if you knew more about the system that we're studying in this example. Okay. So if you want to look at the loadings for each principal component, which are the contribution of each gene to the principal component, ScanPy also has a feature of plotting that, although the aesthetics are questionable. But here are it's the plot of the PC loadings. So on the x-axis, we have the rank from the most contributing to the variance explained to the least. So here's the highest contributor down to the least. And if you zoom in on this graph, you'll notice that the squiggles are gene names. So you can see, CST3 and this whole set of genes, this combination of these set of genes are contributing highly to the first principal component, whereas principal component 2 is determined more by this different set of genes and so on. So we can also scatter in the principal components of all the genes and see if this is vastly different than the principal component projection of only the HVGs by applying the same set of transformations to that object that we saved as normalized data rather than feature selected data. And let's see what we get. Okay, so here, I think this is a good sign. We see that our highly variable gene doesn't drastically change the shape of our raw data projected into principal component space, at least for the first two PCs. And it's not quite identical, which is also a good sign that our code is working. There are small differences, but not large. And we can also look at how the loadings might be different. Well, I'm not going to bring that up because it's quite hard to read. So can anyone think of other ways about how we can quantify the similarity of gene contributions to PCs if we want to compare what different transformations or normalizations or filters might do to our data? So say we have two different objects like we do here, like normalized data versus feature selected data. How can we compare the similarity of the gene's contributions to the PC loadings? Bill? One way to do that would be by taking the dot product or the inner product of the two first PCs, and this allows us to compare whether or not they are determined by a similar set of genes in a similar weight kind of way. So if the two vectors are heading in similar directions, you would get a positive value for the dot product, whereas if they're totally different, completely orthogonal, in fact, they would be 0. And well, if your two vectors are going opposite directions, they would be negative. So you can apply this to kind of to all the sets of principal components across different ways of transforming the data to try to see systematically how different pre-processing steps might affect the loadings of your principal component space. Okay, so now we come to the question of how many principal components do we want to use when we cluster our cells? And the standard approach is just to visualize the scree plot and choose by eye what we think is a good number. So here's the scree plot. And here it plots the rank of our principal components from 0 to however many we have, which is a number we set in the code, and the amount of variance explained. So here's a ratio, and principal component 1 is explaining the most variance, and then it decreases kind of after principal component 8 or 9 or 10-ish. So I find this kind of unsatisfactory when I want to compare how different procedures perform. So what I did here was write a function that can determine the elbow point more systematically. So I apologize. This is a lot of Python code to digest in a short amount of time. But what we do here is just fit a PCA to the data and then find the point at which the derivative no longer increases. So this is the elbow dimension of our data. And in our function, it will return to us our new data, the number of PCs we use, and our explained variance. And so here's a little bit of a detour about how to extract your cell by gene data from the ScanPy annotated data object and to create a new pandas data frame, which is a little bit more of a routine object that people might have more familiarity with coding with. So in this way, it can make applying operations to your data a little bit easier. So in order to pull data out of the ScanPy object, you can create a pandas data frame and convert the X array, the X sparse array, to a full array. In this case, we're just using the highly variable data. And then we can name the rows with the observation names, which are the cells, and we can name the columns of our new array by calling the variable names, which are the genes in our HVG data. Okay. So to define our function, this won't run it. We can just execute, load it this way, and then create a new frame of data. And then to use a custom function in Python, we simply assigned what arguments we want, what we want to name the arguments that this function outputs, which in this case is a list of 5 different arguments. And we apply this function PCA elbow fit to our pandas data and output the dimension, as well as a few other things that we want to use for PCA in a more rigorous sort of way. So let's check out how our visual hypothesis did. This might take a little while to run on your computer. So the dimension of this elbow fitting function, which is that need tool that finds the knee instead of the elbow because of which way it's oriented, but same idea, tells us that the number of principal components we want to use is 14, not 9 or 10. So, okay, that seems reasonable to me. So we can then subset our data into a new frame that has the cells and their PC loadings. So in Python, we have this thing called list comprehensions, and we're going to name our columns by their PCs, which is something we output by this PCA elbow function. So this will create a vector of strings that are our column labels for each principal component. And we can save the data by creating this new name of the matrix and then using the command .toCSV if you would like. I apologize, lots of code. So another useful command is printing the shape of your data, which you can do with this Python command .shape. And this can confirm that we're doing what we think we are doing in our code. And we see that we have a new data matrix that has 2,673 cells and 14 columns because we wanted to include 14 principal components in our dimensionality reduced basis. Okay. Playing music. Interesting. So where were we? We can plot a new scree plot based on our own PCA function because that old one did not look so great with the decision to use words as the data points in the matrix. So here, using the output from that function that finds the elbow dimension, we can plot based on how many PCs we used, what the explained variance ratio was in a little bit more aesthetically pleasing way. And you can see it's kind of continuous, but nonetheless, if you zoom in, we can check out to see whether or not 14 does in fact seems to be kind of all right. Yeah. So if you would like to save these figures, there's a matplotlib function called savefig, where you direct the axes to the path and give it a name. Okay. Now, back to ScanPy in your typical tutorial. So we've gone through all the steps of the preprocessing before clustering, but we'll talk about clustering in a lot more detail tomorrow. What we will do now is just a brief visualization demo. So the first thing to do when applying the UMAP or t-SNE algorithm is compute the neighborhood graph. So here I've chose the principal component feature selected data as the input, and 10 neighbors to use to construct the graph, and the number of PCs that we're going to use to initialize this neighborhood projection is 14. Actually, I think this is done in the PC space. It's not an initialization, sorry. Okay. So neighborhood calculations can take some time. And UMAP and t-SNE algorithms also take some computational time. But after we do these steps, we have enough information to execute the UMAP function on the PC HVG data. Neighborhood should now be one of the observation names. So if we just print the PC HVG data in our Python console, it will tell us a little bit about all of the stuff that we've generated. So as you can see, it's quite a lot of statistics that are organized according to whether they are cell observations, gene observations, or kind of global observations. We have our neighborhoods, our PCs, our log transform counts, etc. And okay, to visualize the data with UMAP, just execute this command. And this will compute the UMAP coordinates. To actually visualize the data, we need to use the ScanPy plotting module on the UMAP observation, and we can color it by as many different genes as we are interested in. So here, let's make 3 different UMAP plots colored by these 3 marker genes that are known to be important in immunology, and see what our data looks like. And we can see that we do have these kind of distinct and similar distributions of marker genes in different kind of populations, while this gene PPBP is only expressed in a very few number of cells. We can look at how that compares to the t-SNE representation, performing the same set of steps. In this case, t-SNE requires PCA initialization. So we have to indicate the number of PCs we want to use. So let's use 14 again. Okay. And what you can see here is that in our PBMCs, we have kind of similar 3 kind of distinct regions that fall out using both UMAP and t-SNE, given the input parameters, but there might be slightly different shapes. Okay, so now what we're going to do is break out into smaller groups, and I'm going to challenge you to do some coding on your own. So as we've gone through this many lines of code. I'm sorry. We've created the following annotated data objects. So there's an AData file that has the raw cell by gene count, quality control data. You should notice all of these in your variable window, and you can print them all by just typing their names in your console. So AData, we have, this object with the full number of cells and the full number of genes. And we can see how we've changed the data as we've gone through this. So in the code, there are some simple tasks. The first one is to read your raw count by cell, matrix, and pandas and try to perform some calculations using Python commands, NumPy or pandas on your own to calculate the fraction of non-zero entries in the matrix. And then there's a few other coding activities listed below. And I encourage someone within each group to screen share and kind of discuss your troubles and help each other along the way. And also I'll go through each group to see how you are doing and answer any questions. Okay. So I'm sorry, I do not see any of these notifications for the chat. Okay, so Sophia, the link to the day one materials, yeah, I can drop that in the chat. Is there a way to run Python IDE interactively on Huffman 2 cluster? Yes, I've heard it being done with RStudio, but I assume you can also do it with Python, but I'm sorry, I'm not super familiar with the Hoffman cluster. If you'd like, I know some people who are, and I can ask them and get back to you. And then what's the difference between principal component 1 versus 2 versus 3? So they're orthogonal axes of variation. They're different linear combinations of genes that can explain the covariant structure of the data. Yes, it's the sample data that we use for this analysis is in the day one materials. Okay, so I'll send that out again. Okay.